fructose. So fructose is the hardest one to get down, so make sure to practice that one. Let's go ahead and move on to the remaining slides. We talked about the anomeric carbon. So in alpha D fructose, for example, on the anomeric carbon, which I'm circling now, the brand new alcohol that's formed, if it's facing down, it's going to be alpha. If it's facing up, however, then it's beta. Here is glucose, and I'll go ahead and I'll use the highlighter for this one. Here's the anomeric carbon. If it's facing up, then it's beta. If it's facing, on this next example, down, then it's alpha. So we do have glucose in the body in the alpha and beta position. Technically, these are what we call anomers of each other, where one of the molecules is anomerically in the up position on its anomeric carbon, and the other anomer, the alcohol group's in the down position. Now, other terminology you need to know, memorize this terminology right here. Pyronoses, Pyronoses, and this is tomato, tomato, depending on how you say it. But I'm going to use a red pen. Pyronoses are just a general category of six-membered cyclical hemiacetal rings. So if the brand new anomeric carbon is a hemiacetal and there are six atoms in the ring itself, including the oxygen, then it's a pyranose, or some people call it a pyranose. Notice in this newly cyclical hemiacetal formation, we have a brand new anomeric carbon that's formed. The anomeric carbon basically produces a brand new alcohol. So we call this a anomeric carbon. So in the top picture, we have the open chain, and it's basically folding up on itself where the alcohol reacts with the aldehyde in a cyclical hemiacetal reaction. And it shows the formation of the two anomers. The beta D-glucose, as the alcohol is in the up position, and the alpha D-glucose, as the alcohol is in the down position. So furanoses, the furious five, these are five-membered rings. That means it requires 5 atoms in the heterocyclic ring, including the oxygen. So these are heterocyclic molecules. So make sure you have those memorized, those terms memorized, as pyranoses and furanoses. And I always think furanose, furious 5, furanose 5. So they have 5-member rings. So here's an example of a pyranose, 6-membered ring. And it turns out that these alcohols are important for 2 reasons. One reason is every time you add an alcohol group to a hydrocarbon, it makes the overall molecule more soluble in water. And considering all cells on the inside of a cell as well as outside a cell are in a watery environment, it's aqueous, then that's a good thing for dissolving these sugar molecules. So the more alcohols you have, then the better, or the more soluble the carbohydrate is. The next reason is this is where all the chemical reactions love to occur, are on these alcohols. Now there are some alcohols that are used more often than others. The ones that I'm highlighting with my pen tend to be used quite a bit. this one, and obviously this one right here. So these are monosaccharide reactions where they react at the alcohol group. In this case, this forms a glucose 6-phosphate. Now, this is how they came up with the name. They numbered from the anomeric carbon, carbon 1, 2, 3, 4, 5, and then they went up to carbon 6. And because there's a new phosphate group attached to it, then it's called glucose 6-phosphate. Again, this is something you have to memorize. Let's go over here. We call this methyl alpha D glucopyronoside. And basically, it's the same thing as a glucose molecule. The only difference is the anomeric alcohol went ahead and it formed a ether that we learned in organic chemistry, or the organic portion. So in this case, here we have a brand new ether, and it's connected to a methyl group. So this is the methyl, and here's the rest of the name, alpha D glucopyronoside. Now, whenever you have any type of ether or another sugar molecule connected at the anomeric alcohol, we now call this a glycoside. A glycoside. Consequently, instead of calling this an ether, biochemists, they love calling this a glycosidic linkage. A glycosidic linkage. That's the fancy biochem term. So, in this case, we have a ether reaction. Whoops, we call this a glycosidic linkage. And it requires two monosaccharide glucose molecules. The first one's going to be the alcohol on the anomeric carbon, and then we have carbon 4 that's going to react. These form a brand new ether, and the fancy biochem term again is glycosidic linkage. Now, when you have this linkage right here, these sugar units are paired up permanently, and we call this a disaccharide. Disaccharide. These are the 3 disaccharides you have to memorize. So know those inside and out. Now, if you go ahead and take a look earlier, in our alpha D glucose, we got the name alpha because the alcohol is in the down position. When it forms the new ether, we have 2 positions that are possible. We can either have the ether in the down position like this, or, and I'll just put R's on both sides. The R's represent the sugar units. Or we can have it like this, where it's in the up position. Now, if it's in the down position, we're going to say it's alpha. And we usually indicate that it's a glycosidic linkage because we say 1, 4. That means it's a glycosidic linkage at carbon 1 to carbon 4. So what I did was I wrote C right above here, and it's carbon 1, the anomeric carbon, to carbon 4. And this is carbon 4 on the second carbon. So we indicate it just as it's written here. Alpha 1, and then you put an arrow there, 1, 4. This is important because at a later time, a cell may want to break this down for energy, and when it does, the very first thing it needs to do is to go ahead and break this glycosidic linkage into monosaccharide units, so that way it can break down the sugars individually. And certain enzymes need to be present to break down the glycosidic linkage, provided it's in the alpha position. If it was in a beta position, then even that subtle change, the enzyme may not work like it would with the alpha, and you would need a completely different enzyme in order to break down the beta form of the ether, aka the beta form of the glycosidic linkage. So it's very important when you sketch this that not only do you sketch the disaccharides with the alcohol groups in the proper form, but you also sketch the linkage properly. And those tend to be graded right or wrong. So this is the name of the disaccharide maltose, and I will say sketch maltose. And you will have to sketch it just as you see here. Also, this is a alpha 1 to 4. Because originally it was on the anomeric carbon where it was a alpha alcohol, and it's still facing down, so this is alpha 1 to 4. Lactose. Lactose in this one tends to be in the beta position. So here is lactose. Notice you start off with galactose, and it has a glycosidic linkage to glucose. And this is how you need to sketch it. Notice it's always in the beta position. So on the anomeric carbon 1, it's going to be attached to carbon 4. Notice it's a beta. Sucrose is the hardest to draw, and this one requires a lot of practice. So in this case, here you have glucose, and here you have fructose. Sucrose, yeah, pretty cool stuff. So sucrose is a very sweet sugar, and it's going to be. So what would this one be? You're right. So carbon 1, carbon 4, alpha position. So it would be alpha 1, 4. So these names, their glycosidic linkage positions, and the overall structure you have to know inside and out, as well as some industrial properties, such as lactose is found in milk, sucrose is found in plants, sugarcane, and let's move on. Polysaccharides. Now, notice this is a repeating. It's a repeating. Whoops. Man, I hate this pen. So it's a repeating, before I was rudely interrupted, glucose. So having memorized the Hayward structure of glucose, it's going to be alpha 1 arrow 4. That's a glycosidic linkage for all of these. And they'll all be facing down, just as it's written right here. I may ask you to write 3 or 4 glucose units in the polysaccharide, or I'll just leave it up to you to decide. And I'll be looking for 3 or 4. And again, these extend out thousands, but you only have to write 3 or 4. Types of polysaccharides. This one, starch is a general category of glucose, polysaccharide glucose units. We have unbranched. That means they're in a nice straight chain. So think of your traditional organic chemistry. So in this case, what I'm drawing right here is a unbranched alkane. And then if I were to go over here, I'm drawing a branched hydrocarbon. And you can think of the same concept when they talk about branched polysaccharide. So in this case, we have our traditional straight chain here, and then on carbon 6, that's the carbon that actually extends out on the top. It's going to go ahead and form a glycosidic linkage. So, if it's unbranched, the fancy name is amylose. If it's branched, the fancy name is amylopectin. Amylopectin. And this is, we can usually put iodine in here, and it creates a nice blue color, and that's how you know you have a starch. That's how you know you have the branched starch version present, such as amylopectin. Glycogen is essentially the same as amylopectin. Matter of fact, if I ask you to draw or sketch glycogen, you could just basically draw amylopectin, and I'll know the difference. Basically, glycogen is just like amylopectin. There's just a ton more of these branched glycosidic linkages that are present. You don't have to know the difference. You don't have to indicate that. Simply sketch what you see right here. Same exact thing. You need to draw the glucose units with the alpha 1,4, and then occasionally you'll draw a alpha 1,6 linkage. And let's finish this up. Cellulose. Here's an example of cellulose, and this, if you notice, cellulose is different from our other polysaccharide. Let me go back a couple slides. So here is amylose. Amylose is a type of starch, and basically starch is used. It's basically used as storage, and eventually will be cut into pieces for energy. So even though this is the stored form, its ultimate purpose is to be broken down for energy. Compare that to cellulose. Cellulose is actually meant as a structural component. It's meant for structure. For example, cell walls are mainly made up of cellulose in a plant. So it's meant to provide structure. Now, do we want to break down a sugar unit that is meant for structure? No. Just as we didn't want to break down deoxyribose because it was a structure unit for DNA, we don't want to break down this polysaccharide cellulose. So, what is the big difference between amylose and cellulose, however? Even though they're both sugar units, it's all about the linkage. The linkage. So in this case, cellulose is going to be beta 1 and 4. Compare that to amylose, whereas amylose, using my highlighter, is alpha 1 to 4. So, sketching this is going to be pretty easy. If you memorized how to draw polysaccharides such as amylose, then all you have to do when drawing cellulose is to make sure each of these glycosidic linkages are going to be forming in the beta form, just as you see here. Careful, as these may be graded right or wrong, and not only do I look at the hydroxyl groups, but I also look at the linkage to make sure you're drawing the correct one. Consequently, whenever we eat cellulose, such as grass, it goes right through us because we don't have the enzymes to break down cellulose. There are animals that can break down cellulose. Basically, anything that eats grass has the enzymes to break down cellulose, which makes up the cell walls, which you would find in grass, or the cells of grass. Consequently, cows, for example, they have the enzymes for breaking it down. And once the cellulose is broken down into individual individual monosaccharide units, then then it can just continue on. We, however, don't have those enzymes, so it's used as dietary fiber whenever we have it in our body. Basically, it just goes right through us. And that's the end of this commentary.